Today's lecture is on antibiotics. And if we were to dissect that word, anti means against, biotic means, or bio means living. So it literally means substances, I guess, that are against living things. Today you're going to want to have the notes that are outlined on the Schoology page. You can either open them up, print them, and fill them in by writing, or you can open the notes on your Chromebook and then just type them and then print them after that. Whatever makes you happiest. To begin with, we have to understand some terms to make this lecture make sense. The first term we have to understand is pathogen. A pathogen is a disease-causing agent. Again, a pathogen is a disease-causing agent. And we use the word agent because there's many examples of different kinds of things that are actually pathogens. They can be bacteria, viruses, fungi, protists, worms. Any of those things can cause an infection to occur. Antibiotics then are a natural product that kills other living organisms. Again, it's a natural product that kills other living organisms. And antibiotics actually originate in nature. So there are plants that will give off antibiotics or chemicals through their leaves or whatever to keep bacteria from growing on them. There are plants that give off chemicals in their roots that prevent other roots from growing in from other plants. There are fungi that give off antibiotics to prevent bacteria from growing in their space. So antibiotics in nature are really about an organism's attempt to kind of defend its own space so that it can continue living. Where it says pathogen growth, then we want to know that pathogen growth is exponential. And so I'm going to watch this tiny little video here to show you that. So there's a few bacteria. Oh, now there's more. Now there's a lot more. And now there's a ton. And so that's just trying to demonstrate exponential growth, how a few become a few more, and then a lot and a lot and a lot and a lot, and so on and so forth. All right, let's look at the history of antibiotics then. So we want to understand that prior to 1880, we didn't really know a lot about what caused infectious disease. There were theories that you got sick because someone in your family was bad, or you were bad, or generations ago. That disease was really a punishment. Well, Pasteur was one of the early scientists to put together the idea of germ theory, that infectious diseases are spread by pathogens, not by some other magical form. But you can see we go from 1880 to 1928 before we have the first antibiotic. And this was done by Fleming, and his discovery was in penicillin and mold. A little bit more about his story. I like when we can find firsthand references to things scientists actually wrote. And Fleming, these were some notes. When I woke up just after dawn on September 28, 1928, I certainly didn't plan to revolutionize all medicine by discovering the world's first antibiotic or bacteria killer, Fleming would later say, but I guess that's exactly what I did. Fleming's discovery was actually kind of an accidental discovery. So he was working on his lab with a bacteria called Staphylococcus, which now you know is a bunch of little round bacteria that are clumped in a group. He went on vacation, he just kind of threw a bunch of his leftover plates in the garbage can. When he came back, they were still there, and what he noticed was, this greenish teal bacteria did not grow over the whole plate. It's really not teal in real life, I just made it that color. But anyway, he would have expected that bacteria to completely cover the plate as it was undergoing exponential growth. But he found some gaps in there. And so as he studied what was in those gaps, he found a fungus or a mold, and it was excreting something that prevented the bacteria from growing. So kind of the first of the antibiotics. There are many things that are discovered by accident in science, and among them was Viagra. As we know, Viagra is used to treat erectile dysfunction, but that was not what it was designed for. It was designed for patients with angina, which is heart pain. So it was designed to treat heart conditions. And as a side effect, many of the patients recorded or shared that they were having erections. And so it became a better drug for erectile dysfunction than it was for heart treatment. All right, back to our story. So Fleming had his fungus that was producing an antibiotic, but he didn't think it was really such a great idea. So he thought the bacteria would become resistant soon and they wouldn't be worth anything at that point. It was really tedious to grow and collect, and he thought it was broken down by the body before it might actually kill the bacteria. So he was working on this, or the U.S. Army was working on this. They kind of didn't really do a lot with it for the reasons that he stated. In 1942, there was a fire at a Boston Army facility, and there were lots of injured soldiers who had burns. And burns make the body very susceptible to infection because your skin is this awesome protective barrier. Since they had many soldiers who were probably going to die anyway, they decided to try Fleming's antibiotic. And it worked. And so chemists continued working on it and making it into, some of the products that we see today. The 1960s came along and the U.S. Surgeon General declared there would be no more infectious disease because we would learn to control them. That person was very wrong. All right, back to your notes then. How do antibiotics work? Well, first of all, you have to figure out how to get them to the site of the infection. So in the case of skin, that's really easy. You're simply going to put it on top of the skin, and there it is. It's right next to the bacteria. It can have its interaction and kill them. If it's some kind of a medicine you're going to give people, that's way more complicated. You have to figure out how you get it past the digestive system. So the pill has to not dissolve in the mouth, go down through the esophagus, get into the stomach, probably maybe start digesting there, but not completely, pass into the small intestine. Then you got to be able to get it into the blood. And then finally in the blood, it's got to be able to get into the target cells. So those are, three kind of hurdles that are very challenging to overcome. How do antibiotics work then? Well, there's probably 4 or 5 different ways, but I picked the 2 I thought we would understand. First of all, you can, basically it has to interfere with the process. The bacteria needs to stay alive. So for instance, if I wanted to kill a plant, I could put water in it, I could put acid on it, I could keep it out of the sun, I could take all the soil away. So again, bacteria or antibiotics have to work by interfering with some process that's critical to keep the pathogen alive. Often that's an enzyme, either interfering with it or blocking it or whatever, because enzymes are important in keeping cells alive. All right, let's look at our 2 ways. One is to stop cell wall synthesis. And that's how penicillin works. So basically a cell is getting, bigger and bigger. And if its cell walls are not getting bigger and bigger, they're getting stretched and stretched and stretched until eventually this happens. So we're going to watch that again because it's a whole 9 seconds. But you'll see the bacteria growing and getting longer and longer. And then basically stretching and breaking like a balloon. Another way that antibiotics can work is by stopping protein synthesis. And we remember from our lessons on DNA that DNA opens up, makes messenger RNA, and that's the instructions for making proteins that keep cells alive. And so that's how tetracyclines would work. All right, now let's look at which drug would you use and how much would you give your patient. So basically, if you look at this, and this is bacteria growing on the plate, you think A, B, C, or D would be most effective at killing that bacteria. Well, hopefully you picked B. And the reason that you'd want to pick B is because its zone of inhibition, the space around the discs, so these different discs have different antibiotics on them, it prevents the bacteria from growing in the largest region. So where it says zone of inhibition, I'm going to ask you to write the words area where the drug keeps the pathogen from growing. Again, the zone of inhibition is the area where the drug keeps the pathogen from growing. And the bigger that zone of inhibition, the more effective the antibiotic would be. So if it's completely ineffective, then the little bacteria would just grow right on top of it because it wasn't doing anything to destroy them. So once you've figured out, okay, which antibiotic is most effective against this type of bacteria, how do I figure out how much to give to my patients? And so we're going to start with toxic dose. Now, let's say you're in college and you really need some money. You could volunteer to be part of some scientific research, and you would be part of a toxicology study. Basically, they'd give 1/4 of the group 1 milligram of this drug, 1/4 of the group 2 milligrams, another group 3 milligrams, and the last group 4 milligrams. And then everybody writes down over the course of the month they're taking the medication, how do they feel? So if they figure out the people that get 1 milligram, they feel just fine. The people getting 2 say, oh, I have occasional headache and maybe a little bit of nausea. Group 3 says, I have a constant headache, I've had diarrhea for 28 days, and I have double vision. All right, we know then that the toxic dose is between 2 and 3. 2 gives you a few side effects, but 3 is just way too much. So please put toxic dose is the amount that causes negative side effects, or better, maybe too many negative side effects. The therapeutic dose then is how much do you have to give to your patients to make them well. So is it 1 milligram? They go, nope, that didn't help me. The group who got 2 said, yeah, I feel better. The group who got 3 or 4 or whatever, ooh, we feel really good. So trying to figure out what's the minimum dose you can give people therapeutically to make them healthy, but not cause a lot of side effects. So the therapeutic dose is enough to make your patient well or healthy. And often this is a balance. You might be taking a drug that's helpful to you, but has toxic side effects. And you have to kind of then choose whether it's worth those side effects. So if you think about people with cancer, that's a decision that they make in terms of the drugs they choose to use. All right, let's talk about different cleaners. And so antiseptics basically destroy cell walls. So once you've broken the wall, the stuff's going to leak out and the cell's going to die. So it's like bombing it from the outside. So antiseptics work from the outside, moving their way in. Disinfectants really do the same thing, except they're way too strong to put on your skin. So bleach is a great antiseptic, but it's too harsh to use on your skin. So you use disinfectants on non-living surfaces like countertops and equipment. Antiseptics like hand sanitizer, alcohol, iodine can go right on your skin. Antibiotics then, they work the opposite way. They work by getting inside and destroying from within the cell. Last slide here. Pathogens, how do we get antibiotic-resistant bacteria? Well, basically, they find a way to combat, I guess, the antibiotic. Maybe they pump the drug out faster than it's going in, therefore it doesn't have any effect on them. Or they have an enzyme that helps them detoxify or break down the antibiotic before it damages the cells. So because enzymes are products of the DNA that we have, they build those enzymes because they're proteins. You can pass that trait on to your offspring. And that would be how bacteria pass that trait on, creating superbugs. and superbugs are just resistant to the antibiotics we have.